Welcome to Biology at Priyajit, my dear students. Today we will explore apoptosis, mechanism, cytological features and its differences from necrosis. Apoptosis is basically a genetic programmed process by which cells die in an orderly and controlled manner to maintain tissue balance and normal development. Understanding how apoptotic pathways work, how the cell changes during this process, and how it differs from necrosis will help you appreciate its role in health, development, and disease. What is apoptosis? Apoptosis basically programs cell death. All living cells, whether it is animals or plants, even bacteria, are mortal. Normal genetically controlled cell death is called apoptosis or programmed cell death or PCD. It is very much essential for normal development and maintenance of tissue homeostasis. Apoptosis or programmed cell death is basically different from accidental cell death like necrosis. So defect in apoptosis causes uncontrolled cell survival which promote cancer and genetic instability. So cell in the body when it undergoes internal breakdown, fragments recycled or destroyed. So how apoptosis helps keep the body healthy? It replaces old cells, it strengthens our immune system, it gets rid of damaged cells, it stops damaged cells from multiplying and support overall cellular balance. Cellular events during apoptosis is a physiological response to death signals and absence of survival signals. Sequence of events: chromatin condenses and moves toward nuclear membrane. Nucleus and cytoplasm shrink. Plasma membrane remains intact. Cell breaks into small membrane-bound fragments called apoptotic bodies. These are phagocytosed by macrophages or neighboring cells. No inflammation occurs. These are the cellular events during apoptosis. What are the cytological features of apoptotic cell? Cell shrinkage, loss of water and cytoplasmic volume, chromatin condensation, dense crescent-shaped masses near nuclear membrane, nuclear fragmentation, which is also known as karyorrhexis, nucleus breaks into pieces, membrane blebbing, bubble-like protrusions without rupture. Apoptotic bodies, membrane-bound vesicles with organelles are formed. No inflammation, it contents not released into the tissues. Mechanism of apoptosis or Caspase pathways, mitochondrial or cytochrome pathway. It involves BCL2 family proteins. Releases of cytochrome C causes APOP1, activation of cytochrome 9, activation of caspase 9, and which also activate caspase 3, which is the executioner caspase, and it leads to execution of apoptosis. TNF, tumor necrosis factor, it's a death receptor pathway. TNF ligand binds to death receptors. It activates caspase 8, which promote caspase 3 and ultimately causes apoptosis. Granzyme B pathway, cytotoxic T cells release granzyme B and it directly activates caspases which promote apoptosis. Role of CED genes. CED genes is the caspase or the enzyme found in Caenorhabditis elegans death protein. Caenorhabditis elegans death, that is the origin of the CED. It was identified in C. elegans or Caenorhabditis elegans and Ederia. CD3 and CD4 are pro-apoptotic genes and respectively protein, and CD9 is the anti-apoptotic which prevents apoptosis. CD9 blocks CD3 and CD4 activity. So these genes regulate all programmed cell death. Caspase, uh, caspase is basically cysteine specific aspartase. So it is a cysteine protease enzyme. It cuts protein at specific aspartic acid residues. Its active site contains the amino acid cysteine, and aspartic acid it cuts on the site of aspartic acid. It causes organized dismantling of the cell. It leads to nuclear breakdown, cytoskeletal collapse, and formation of apoptotic bodies. Importance of apoptosis: it maintains tissue homeostasis, it removes unnecessary embryonic structures like tail in tadpole, it eliminates damaged and mutated cells, it acts as a natural anti-cancer mechanism. Necrosis, uncontrolled cell death due to infection or toxins and trauma. Cell membrane ruptures, cellular contents leak out, causes inflammation and tissue damage. So this is a normal cell. It undergoes necrosis and apoptosis. It disintegrates, swelling when irregular. It condenses and causes fragmentation. So this is the differences table showing the differences between apoptosis and necrosis. Apoptosis causes shrinkage of the cells, blebbing of the cells, DNA fragmentation occurs. Necrosis causes swelling, membrane rupture, and ultimately inflammation. So as we conclude, remember that apoptosis is a carefully regulated process of programmed cell death that maintains tissue balance and removes unwanted damaged cells. So understanding its mechanism, cytological features and differences from necrosis helps you realize how the body controls cell survival and prevents disease. Keep exploring the beauty of cell biology, the beauty of cellular regulation. Thank you.